Welcome to Inert Chemistry. In today's video lecture, we carbapenem and monobactams. It is a very important class of beta-lactams, and you know that we in the previous some videos we start discussing about beta-lactam drugs, and this is the fourth lecture of this series. In today's video, we discuss carbapenem and monobactams. We see the chemistry, mode of action, metabolism, and subclass. Carbapenem. Carbapenem is one of the important class of beta-lactams, and it contains 4 to 5 subclasses. Among these, we discuss imipenem. It is a very important drug. So let's first discuss the chemistry of carbapenems or imipenem. This is the basic structure of carbapenem. Among like all the beta-lactams, it also contains a beta-lactam ring. You know that the beta-lactam ring consists of 3 carbon atoms and 1 nitrogen atom. So this ring B is a beta-lactam ring, and it is the characteristic of beta-lactams. It is present in penicillins and cephalosporins that we had already discussed, and same here it is also present in case of carbapenems. In addition to this beta-lactam ring, there is a 5-member ring, and sulfur is attached to this 5-member ring. Similarly, here hydroxyl side chain is attached to this beta-lactam ring. If we talk about the mode of action, then carbapenems are the antibacterial drugs that work by inhibiting the cell wall synthesis or function. Metabolism of carbapenem, or specifically imipenem. Imipenem is a carbapenem antibiotic that is used to treat a variety of bacterial infections. It is administered intravenously and it is rapidly metabolized in the body. Imipenem is metabolized primarily by the enzyme dehydropeptidase 1 in kidneys, which convert it to a less activated active metabolite that is called dehydropeptidase imipenem. Here we see metabolism in somewhat detail, and in previous videos we studied the metabolism of drugs, and we had learned that during the metabolism 2 types of metabolic reactions are occur: phase 1 metabolism reactions and phase 2 metabolism reactions. During the phase 1 metabolism reactions, oxidation, reduction, or hydrolysis of drug occur, while in phase 2 reaction, the drug is conjugated with some other molecule so that it is made more polar and more hydrophilic. And remember that the major organs that are involved in the metabolism of drugs are liver and kidney. So if we talk about the metabolism of imipenem, then in hepatic or liver, there is no or we can say that there is very little metabolism of imipenems occur in the liver. The major metabolism of imipenems occurs in kidneys by the help of enzyme dehydropeptidase, and this dehydropeptidase enzyme, they causes the hydrolysis of imipenem and convert it into a less active metabolite that is called dehydropeptidase imipenem. Now we see monobactams, or it is the only class of monobactam It is the only subclass of monobactam asteronum. Here you see the chemical structure or chemistry of monobactams. It contains only a beta-lactam ring, or we can say a substituted beta-lactam ring. This 3 carbon and 1 nitrogen containing ring is beta-lactam ring, and on this beta-lactam ring various substituents are attached. It also contains R group. If we talk about the mode of action of beta-lactam of monobactams like asteronum, they are like all others beta-lactam. They work by inhibiting the bacterial cell wall synthesis. Here is a bit detail about the metabolism of monobactams. The monobactams are primarily work on penicillin binding proteins. The penicillin binding proteins are actually the proteins that are involved in transpeptidation reactions in during the formation of cell wall of bacteria. There are various types of penicillin binding proteins, and the monobactam mainly acted on penicillin binding protein 3 and deactivated them. Thus, the transpeptidation reactions of cell wall synthesis do not occur, and thus the cell wall becomes weak and fragile, and it loses its structure, and due to the osmotic pressure the cell remains no more, and thus it causes the death of bacteria. Now we see the metabolism. Asteroderms like monobactams, they mostly undergo phase 1 metabolic reactions in liver, and in liver hydrolysis of these monobactam drugs occur. The phase 2 reactions are that involve conjugation, glucuronidation, sulfonation. They are typically not significant pathway for asteroderms. The asteroderms only undergo phase 1 metabolic reactions, and particularly hydrolysis occur, and phase 2 reactions are not very significant in case of asteroderms or monobactams. This is all about today's lecture. Thanks for watching this video. We will soon come with another video.